Hello, cloud community of BioEssay readers. It is my great pleasure to welcome you together with two of the lead authors of the study that inspired me to write this recent review on the newly discovered harvestmen relict eyes, eyeing for their functions. And so our guests are Dr. Prashant Sharma, University of Wisconsin, and Dr. Guillermo Gaynet, who is currently at the medical school Harvard. And they were the major forces on this paper that came out early this year. The vestigial organs alter fossil placements in an ancient group of terrestrial chelicerates. So one of the things that struck me when I read the title and then followed the story actually into the data, that it doesn't talk about harvestmen or contemporary species, but it sounded like the premise was to answer a question about a fossil species. So if you could give us a quick background, Prashant and Guillermo, from which questions and from which areas your research trajectory actually comes from.
Sure. I'll kick it off and then I'll hand it over to Guilherme. So the beginning of this investigation takes us back through the fields of taxonomy, phylogeny, and paleontology. For a long time, it was thought that daddy longlegs, or Opiliones as the group is formerly known, possesses only a single pair of eyes. And this was the conventional thinking for a very long period of time. And this is how we described a lot of our species, with the understanding that this one pair of eyes was likely homologous across the group. About 10 years ago, in early 2014, we encountered a fossil from a Carboniferous siderite nodule, which appeared to have what would seem like traces of two pairs of eyes. So the idea there was that this animal retained both a median pair of eyes and a lateral pair of eyes, but the evidence was indirect until Guilherme started to look at living species. And I'll let him take over from here.
Yeah, so I think when I started this project, I wasn't thinking too much about the four-eyed fossil. My idea was to study the molecular basis for the reduction of that visual system, thinking about some parallels in cave species, the Mexican cave species, and these sort of models. And when we started looking at the gene expression, then the story started taking shape and was a very pleasant surprise.
So then, of the two of you, who considers themselves more of a harvestman or daddy longleg species expert, aka taxonomist?
Oh, Prashant for sure. Prashant is a great systematist for
I think
harvestmen.
I think my only advantage here is that I'm older. So Guilherme has found some really, really cool species of daddy longlegs. He's done some wonderful SEM or scanning electron microscopy surveys of their sensory organs across the entire clade. And this was a large part of his undergraduate and master's work in Harvard and in Germany before he joined my lab. But I started out as a classical taxonomist, mostly working on blind species of these animals.
Which means you should have no problem identifying the species on the next picture.
Yes, we encounter these kinds of leobunines very frequently. So this is a member of the largest family of the sclerosomatids. This is the one that you're most likely to encounter if you're in North America or Europe. So this is a very, very common species. Yeah.
So then this is to give our readers who have maybe less time to undertake walks in the woods a real or a almost close-up impression of how a eupnoid species or a generic one looks like. So the leg to body length dimension is, of course, very impressive. And then we have here this central pair of eyes. So these are the median eyes. And this animal, based on your studies, of course, should have additional eyes which are cryptic and therefore not visible. And so one piece of evidence is shown here in a staining that you kindly shared with me but didn't publish yet. And so that is a triple staining that features a rhodopsin in green, the transcription factor PAX2 in red, I think, and then there's also the transcription factor I is absent. If Guilherme or Prashant, would you be willing to run us through the most important aspects and where we find the cryptic eyes?
Sure, I think I can take from here. I think the most important aspect in this figure is the colocalization of the yellow expression. So eyes absent is a general, very conserved eye gene across many species. So it's expressed in the main eyes that we knew were there, which are these two oval structures right on top of the image. But they colocalize with these green dots, which is the expression of opsins. So opsins are proteins necessary for the transduction of light. So it's a good readout for cells that are most probably photoreceptors. So the overlap of this eye transcription factor with a structural visual gene was a very good hint that we were looking at a visual organ. And then in our paper, we did a set of other experiments to test this hypothesis that we saw from this initial expression.
Very helpful. Thank you, Guilherme. So that means this is the type of experiment you, of course, ran on your model organism, aka Phalangium opilio, whose eyes we're seeing here. And so these are my cartoonish sketches of different distributions of central eyes, median eyes, and then including the lateral eyes. So now from your perspective, that is an adequate representation of eye arrangement diversity in the harvestmen? Or are we missing some extreme variation not included here?
But I think that about covers it. So within the mite harvestmen, within Cyphophthalmi, the sister groups of the rest of the harvestmen, when eyes are present, they're always associated with these promontories that we call ozophores, which are shown in your diagram with the yellow marks. And those ozophores and those associated eyes, the ozophore is always present. The eyes may be present in species of two families. And those can occur in slightly different positions on the head. They're usually projecting kind of dorsolaterally, but they can be a little bit more dorsal or a little bit more lateral. With the remaining daddy long legs, we will see conditions kind of like what is photographed here, where the eyes are very close together on top of the head, or they can be very far apart. And that entire promontory associated with it, the ocularium, can be very flat. So this was part of the reason why there was so much back and forth about whether all of these eyes might be homologous, because there was considerable overlap between the positions and the degree of outgrowth of the structures that hold the eyes.
But I think thanks to your work, we can be confident that the homology calls are now settled in general for eye diversity in the harvestmen, or you're still getting discussion asking for more investigation of the cyphophthalmi?
I think the major point of consideration here that we will often receive is whether we have investigated if vestigial median eyes are also present in mite harvestmen. And mite harvestmen are hard to obtain. Their eggs or embryos have been described maybe once in the thesis of a French scholar named Ghiberti in 1964. And since then, nobody's really looked at mite harvestman eggs. After the study was published, I went to Savannah, Georgia, and now I keep a colony of them. I'm still waiting for eggs. It's been the good part of a year, and I haven't gotten any, so they're tricky. But if ever we manage to get that to work, our hope is that we might be able to survey for these as well. Yeah, so there's still some missing pieces of the story across the group.
So we are going to see more papers on that topic in the next 2 years, or maybe in 5 years from now.
So if we get that lucky, I don't know. Yeah.
Well, what I can tell you is I am looking forward, so the pressure is on. I would like to congratulate you on your fantastic work, and I would like you to thank you for your precious time, and wish you all the best from here.